All right, thank you for having me here. I'm Marcus. I just started a lab at NYU School of Medicine. I was also a Louis Sigler Fellow with anesthesia at Princeton before going there. And what my lab is interested in is how proteins interact with their target. So I'm actually an experimentalist, and this talk will be a lot different from some of the talks that we've already seen. So we're interested in protein interactions, so protein-DNA interactions or protein-protein interactions. We might be engineering those interactions for a synthetic biology application, or we might be trying to characterize those interactions for more systems or network applications. Either way, we're interested in the nuts and the bolts of these sorts of interactions. And today I'm going to talk about trying to find functional but low affinity interactions. And the reason for this is we're finding more and more in biology that there are very meaningful low affinity interactions. So for example, Mike Levine recently published a paper about what he called suboptimal binding sites and how those relate to transcription. There are a number of protein-protein interactions that have been reported that are involved in signaling pathways, where turnover is very important, so high affinity interactions can actually slow down that turnover. But my favorite example comes from the labs of David Stern and Richard Mann, where they're talking about homeodomains, which are very near and dear to my heart. And it addresses this paradox in the homeodomains. So there are many different homeodomains, and they're very important in development and determining the body plan. But for the most part, whether they're binding as a monomer or in a complex, they bind very, very similar sequences. So how is it that these proteins are able to control very different functions and very different developments when they're all binding to the same things? And what Dr. Mann and Stern found was that the high affinity interactions, all of the homeodomains are binding to those sequences. It's the low affinity interactions that are discriminating between an interaction with one homeodomain versus another. So it turns out that the very functional and very important interactions can be the low affinity interactions. So if our techniques to characterize these interactions are biased towards affinity, we're losing a huge picture of the puzzle. And this becomes evident when we look at many of the common techniques that we use to characterize these interactions, like CLAX and phage display are techniques that are used that use multiple rounds of selection or multiple rounds of enrichment that lead to a consensus, and ultimately these are higher affinity interactions, so we're losing lower affinity interactions. And then there are other methods like hybrid assays and protein binding microarrays where you set some threshold and you're going to lose the interactions below that threshold and just recover what's above that threshold. If you set that threshold very low, it can be difficult to discern what is a functional and a non-functional interaction. So just to sort of sum that up, if we're using a technique that enriches for something, we are leading towards high affinity interactions. If you're using a technique that sets a threshold, it can be hard to discern functional from non-functional interactions. So what my lab works on is an old technique that we try and modify. We use a hybrid assay, and we use a bacterial hybrid assay that I've been working on since I was in graduate school. So to explain what we're trying to do, I'm just going to give a review, a small little review of hybrid assays that I'm sure you're all familiar with. But just to make some points, the idea behind just about any hybrid is that you have some gene that is your reporter gene, and this is something that we measure, or that we can measure. And we're trying to make the output of this gene a meaningful representation of the interaction that we're interested in. That might be a protein-DNA interaction, or it might be a protein-protein interaction. So in this example, we have a known protein-DNA interaction, a known protein-protein interaction, and we ask the question, do these two proteins interact with one another? If they do, this whole ball of stuff gets recruited to the promoter and turns on your reporter, which is, again, something that you can measure. Now, we have all kinds of different reporters that one might use. There are selectable reporters like His3 and Ura3, and the beauty of a selectable reporter is you can screen through very large libraries to find potentially rare variants that actually interact with one another, because you can make survival of these cells dependent on that interaction. So it's a great screening method. Alternatively, scalable reporters like GFP, they have different levels of output, so the amount of that output can tell you something about the strength of the interaction that you're interested in. Now, you can do hybrid assays in all kinds of cell types. We like bacteria because it's very easy to use, it's very fast. I'm probably the only person who thinks that yeast are too slow. But the main reason that we like bacteria is the 10 to the 9th transformation efficiency. So this means that in a single Eppendorf, I can look at 1 billion combinations. So this level of throughput really lends itself to, the kind of systems level experiments that we're interested in. Okay, so we use a bacterial system, and to explain how this works, I'm going to go back to the very beginning in Scott Wolf's lab where I did my graduate work. And the development of this specific version of a hybrid system, where it started there, was to characterize the transcription factor specificity, or the DNA binding specificity of different transcription factors. So how this works is we express the DNA binding domain, or a factor of interest, as a direct fusion to the omega subunit of bacterial polymerase. That just functions as our activation domain. Then we place a region of random sequences upstream of the promoter that regulates our reporters, which are His3 and Ura3 from yeast. We do this in a bacterial strain where we've knocked out the equivalence of these reporters, so that when we grow these cells on media that lacks histidine, survival is then dependent on activation of this gene, and therefore dependent on this protein-DNA interaction. Now, like I said before, some methods have different thresholds that you can set. We can set our threshold using 3-aminotriazole, or 3-AT, which is a competitive inhibitor of His3, in order to require more or less His3 to be expressed, and then presumably a higher or low affinity interaction. Now, the greatest advantage of this system relative to any other hybrid assay is omega, and I think this is often overlooked, and that is because omega is part of the core polymerase, but it's non-essential. So we can do our selections in a strain where we have knocked out the endogenous omega. So when we're expressing our fusion protein, we're recruiting polymerase to the promoter that we care about, the one promoter that is driving this reporter, without any competition from omega in the cell, because we knocked it out. So other hybrid assays you may be familiar with, it's very common to have to express your fusion protein at such a level that it's actually toxic to the cell. We don't have that problem because we lack this competition. And in fact, I'm showing you here a Western of a fusion protein that we expressed with 3 different promoters. And it's actually this weakest promoter is the promoter that we most often use. So we can really dial back the protein expression level in the system, and we still get a strong output of our reporters. And that just tells you we get a more sensitive readout of these interactions. Now, in practice, what we do is we transform 2 plasmids into our cell line, and we're doing this in such a way that a single member of your library will get into each cell. So the cell purely functions as a compartment. It's compartmentalizing a single member of your library with the protein that you're interested in. If that's a compatible interaction, the cell will survive under selective conditions. If it's not a compatible interaction, the cell doesn't survive. So once you grow them under selective conditions, you recover those colonies, and then you sequence the window of the plasmid that had been randomized, and you can get a representation of what sequences were required for that cell to survive, which represents the DNA binding specificity of the transcription factor. Since Scott has gone on and now characterized over half of the transcription factors in Drosophila using this method, I've done a lot more stuff engineering with this bacterial system. But to give you a sense of the throughput, a technician in my lab can do 25 selections per week, so it's relatively high throughput. Now, something I worked on with Mona Singh, who is not here yet, is we actually flipped this system around because it's on plasmids. It's very easy to do this so that we can change the bait and the prey combination. So it's just as easy to express a library of proteins and select those that are able to interact with the target that you're interested in. And we did that by screening some enormous zinc finger libraries over a comprehensive set of DNA targets and produced the largest combination of zinc finger or positive zinc finger DNA interactions to date. But I'm not going to talk about that today. Maybe Mona will. But what I want to talk about is how we're manipulating this system to now capture the low end of affinity. So like I said before, we have two reporters here, selectable reporters, His3 followed by Ura3. But Ura3 is really just a historical at this point. It doesn't even need to be there for this application. So what we did was we replaced it with GFP with the idea that if we combine a selectable with a scalable reporter, that we could start by screening through large libraries to find functional interactions and then sorting based on the GFP output to get an idea of what the strength of that interaction is. So just giving you an example with protein DNA interactions, what we've done is we just did a mock experiment where we tried ZIF268, a very commonly used zinc finger protein, with a set of binding sites that have known affinities relative to the consensus target. And without any selection, we just grew these cells and measured the GFP output. And you can see that the GFP output is nicely related to the affinities of these different binding sites. But what was most impressive to us, or what we were happiest about, is that we actually are recovering these low affinity interactions down here above the background. So this gives us the first indication that we can use this type of approach to recover low affinity interactions just above the background using this sort of selection approach. Now this, I wanted to put this up here to let you know that we can do this kind of thing with protein DNA interaction, but everything else that I'm going to talk about is protein-protein interactions. So we have converted this system now to look at protein-protein interactions, just like any two-hybrid assay. What we're doing is we take a DNA binding domain with known specificity and express one protein. We take the other protein and express it as a fusion with omega, our activation domain. We've started out by using PDZ domains because these are very well characterized protein domains that bind to the C-terminus of their interacting partners. So there's a mountain of literature for us to compare what we're doing to and validate the way that our system is working. So the first one we're working with here is the urban PDZ domain. Again, there are many publications documenting how urban binds with its interacting partner that we can use to validate the system. So just like the DNA binding interaction, the first thing that we did was a proof of concept where we tested urban with a number of different ligands that have previously been tested to show decreases in relative folds of affinity based on these substitutions in the ligands that the PDZ domain binds to. And you can see we're getting a nice relationship between our GFP expression and the known decreases in affinity of the urban ligand interaction. Again, we're excited that we're capturing these interactions down here that are a significant decrease in affinity relative to the consensus target, and we're still getting this signal above a negative control right here. And this is becoming, it's more evident that this is important because we've tested the C-terminal peptides from known interacting partners of urban. So these are proteins that we know urban interacts with in vivo. We took their peptides, expressed them in the system, and you can see we get this GFP expression that is above our negative control, but it's very low activity. So these are arbitrary units of GFP in the low hundreds sort of range. And if we go back to this slide, you're talking about this range down here. So when we measure what's going on between urban and these known interacting partners, these are very low affinity interactions. And we're just happy that we're able to recover those. But at this point, what we've been talking about are one-to-one interactions that we've set up. What we'd ultimately like to be able to do is screen through libraries. So the first library that we built was completely randomizing the last 7 amino acids, or the C-terminal 7 amino acids of a protein. We have this library, we're testing it, but I'm not going to show you anything that we've done with it, because I think a more informed library that we've built has been in collaboration with one of our collaborators at the University of Toronto, Phil Kim, who created this library by designing over 50,000 peptides that all come from human proteins or different splice variants from human proteins. So it becomes a more manageable size for us to look through and sequence and find out exactly what's going on here. And plus, it's relevant to what Urban would actually bind to in a human cell. So the first comparison that I have here is between a low stringency selection that we did with this library and phage display results. And while you can tell that we're moving towards the consensus sequence that you would find with phage display, we're recovering a lot more diversity here. And I really want to highlight this -1 position, because what we find when we go back to these things that we've tested before is that there are substitutions in all of the known interactions at this -1 position. None of them have tryptophan at that position. And if we go back to this one, you can see, when we make substitutions at the -1 position, these are very low affinity interactions. So that implies that, what we're recovering in this very low stringency selection is a very wide range of affinities, from substitutions or low affinity at the -1 position all the way up to the consensus target. Now, if we want to lead to, yeah?
The difference is between position 7, between isoleucine and leucine. That's interesting.
Yeah. Oh, I'll throw out a caveat here. So we've just started doing these selections. There actually is an Illumina run going on. These things are based off of about 20 to 30 sequences, so that might just be a sampling difference. I can't tell you why the difference would be for phage display, why they're not getting any leucine but only isoleucine. But, maybe we would find more isoleucine. It would make sense that you would find both, really. But okay. So all of these different logos that I'm going to show you are based on 20 to 30 sequences, though we'll soon have, millions of sequence reads. Okay, so if we want to get closer to a consensus, all we have to do is increase the stringency of the selection, and all of a sudden, we get a much stronger interaction. But that's not the point here. Is we want to actually grab the low end of affinity and then tell us what that means. So since this was a selection, we know that these things survive the threshold that we set. We don't know if there's a difference between the peptides that we select. So that's where GFP comes in, is that we can then start to tease out, are there differences in these things that met our threshold? So the process that we are now taking is we do a selection where we provide a very minimal level of 3AT, or a minimal selection. And what I mean by that, or how we set this level, is we take our library without a protein interaction and start with no 3AT and start to increase it until the cells don't survive anymore. So we get to the lowest point possible where, the cells would survive without a protein interaction, and that's the threshold that we set. It's usually about 0.5 millimolar 3AT, which for anything else we've done is an incredibly low stringency. So we recover everything on this plate. We pool it together. We take a sample sequence of that pool, and then we just sort it out by GFP and recover different pools at increasing GFP levels. So with Urban, this is what you get, where we've recovered populations at increasing GFP concentrations. And you can see that we're moving towards the consensus sequence as we look at higher expression levels of GFP. But what's interesting to me is even at this very low level of GFP output, we're still seeing some hallmarks of the Urban specificity. We're still seeing the valine down here. We're seeing an acidic residue followed by a threonine or a serine. But we're recovering a bunch of diversity as well. So we think that that's interesting. But Another thing I wanted to point out was that this population is actually below this GFP threshold that we set. And I think we're doing this for a reason, and that's because this threshold has just been set by some negative control. But when we've looked at, for example, these interactions before, it's not like the expression level is way above the mean of a negative control. It's just barely above that mean. So what we're actually doing is we start collecting populations at the mean of the negative control and move above there. Okay, so where do we actually find these interactions, these low affinity interactions in the populations that we recover? We're actually finding we're finding these actual peptides at low counts in the low and the medium GFP output. We never find them in the high GFP output. So again, when we submit some sort of a selection like this, we're only finding the low affinity interactions in the low GFP output. This is the same when we look at other PDZ domains where you see this increase in or a trend towards the consensus. When we look at a MAG3 PDZ domain, we've scaled up now. We've looked at 12 other PDZ domains. But what I want to make clear about what our ultimate approach will be, we don't have our Illumina results back yet, so we can't actually tell you exactly what is in all of these pools. The point that I want to make is that the comparisons that we're going to be able to make between this starting sequence here and the different pools will be very informative, where we will be able to reduce or eliminate the bias that could be in this starting sample because all of these different GFP outputs would have started from the same place. And the other thing that we're doing is we're looking at different time points. So we look at one time point of GFP output, then we look another 6 hours later, and we're actually we're testing or we're measuring how the GFP level is changing for any given peptide. Does it go from, how quickly is it going from, the low GFP output to the medium low GFP output? And that speed is telling us something about the affinity, or at least that's how we hope this is going to pan out. Okay, so in summary, it's becoming more and more important, or more and more evident, that low affinity interactions are very important interactions in vivo, but most of the methods that we use have difficulty capturing that low end of functional affinity. We hope that by combining these selectable and scalable reporters that we can recover the low end of affinity, a functional low end of affinity, and then by comparing the enrichment of these peptides across different GFP populations, ultimately we'd like to be able to make claims about relative affinities for all peptides that a protein is able to interact with. The people that did this work, I think I went through this very quickly, but that means we have plenty of time for questions. The people that did this work, the people in my lab that actually set up a lot of these systems at Princeton are Elizabeth, Ben, and Denise. Ben is now a graduate student here at Berkeley in Jennifer Doudna's lab. And amazingly, the amount of PDZ domain work that I've showed here was all done in a single rotation by Xu Wei, who is an amazing graduate student. So if anyone has advice about how to bribe someone to join your lab, would appreciate it, because she's awesome. And then Dave has also helped out in setting up this system. So I'll take any questions. Yeah.
In the random libraries, either the nucleotide or the peptide, are guaranteed to cover all the chimeras? Do you have a sense of what sequences you get in the random library?
Okay, so in the original DNA library that we tested in Scott's lab, it's a 28 base pair fragment, and the library size is 2 × 10⁸. So it's nowhere near covering every possible combination. But the way I looked at it was, we are likely, if you think of a 28 base pair region as 16 overlapping 12 base pair regions, you could think of it as overlapping 12 base pair libraries, and we would actually cover that in 2 × 10⁸. So that's my weak way of claiming that we're covering all 12 mers. But no, Scott could have sequenced the entire library by now, because 10⁸ would not back when we first made that library, it was impossible to sequence. But now you could actually sequence 10 to the 8th, you know. So no, we don't actually know. The large 7 amino acid peptide library that we've created, we actually built it to be 10 to the 10th. So it was a very arduous process to build it, but we, you know, obviously we can't actually sequence all of the variants, but we made a huge library.
Okay, and you showed the plot where you can capture the low affinity site. And you know what's the I mean, you showed that the ranking is similar. You know what there is what's the correlation? Is there some transformation from the GFP intensity?
Yeah, we don't know yet. I mean, that's what we're hoping to find, and what it's going to take, like I was just alluding to at the end, it's probably going to take recovering populations at multiple time points to actually figure out, you know, how the population is changing at different GFP levels over time, to tell us how that's really relating to GFP and if there is an exact transformation between GFP and the affinity.
KD values.
Yeah, I don't know. I mean, that would be, you know, the holy grail for me, is to actually be able to produce KD values with this. I think what we will probably end up having is some relative score. You know, like this is some, you know, X factor of the consensus target, or the highest.
Absolutely.
Yes. Yeah.
I have a related question. So you seem to sort the GFP. Why don't you build it? I mean, this might be a very dumb question. Why don't you build it so you can sequence? Like, depends on how many reads of a tag you get. Like the high throughput reporter assay kind of experiment.
You mean sequence?
So you
Yeah.
based on recount.
Yeah, well,
Equivalent of GFP.
oh, I see what you're saying. So get the counts of the different peptides that are in the different. Well, we sort of get that when we do the high throughput sequencing. You know, we're going to get sequence reads for each protein. So I mean, we don't really need to barcode it to get that, right? Like.
But you're trying to sort the GFP level.
Right.
So why don't you just have some kind of paired end so you can sequence the mRNA level?
Oh, I see what you're saying. So we could actually sequence how much mRNA is being expressed rather than looking at the fluorescence itself. Well, we hadn't thought about that. That's a good idea.
So it's not entirely
No. Because, I mean, there's actually one of the issues that we're having setting up the system. By the way, we haven't even come close to publishing any of this, so, you know, don't tell anyone. But, oh, wait, nobody copy this. But, you know, one of the problems that you have is at some point you just max out GFP expression. You know, you hit a maximum. But mRNA levels, it's different, right? So that's a good suggestion that we could correlate it to the actual amount of GFP RNA that's being expressed.
Well, maybe we should take a break and come back. There's one more unknown speaker. Just look a lot like me. And then don't go away, and be aware there's another hall reception at 4, so you can go have coffee now, but you know, there's more food. So thank you again, Marcus.